The pathophysiology of ischemic stroke. Welcome, and thank you for joining us on this incredible voyage to understand the third leading cause of death in the industrialized world, the #1 cardiovascular cause of death in China and Japan, a leading cause of adult disability in the United States, with a total estimated cost to society of $53.6 billion a year. Stroke. Stroke is caused by a transient or permanent interruption of blood flow that leads to ischemia and triggers a complex series of pathophysiological events that causes injury to the brain. The consequences of these cerebrovascular events for the patient may be death or disability, with the loss of productivity and a decrease in quality of life. The lack of effective acute stroke treatment is a major health burden for patients, their families, and society in general. The pathophysiology of an ischemic stroke involves a complex cascade of events that occurs within the brain. The human brain requires a constant supply of oxygen and glucose, which are used for the production of energy. These substrates are delivered through a vast network of blood vessels throughout the brain. A variety of vascular events, including plaque formation and rupture, can create an embolus or clot. An embolus moving through the vascular system can occlude a cerebral blood vessel, thereby reducing the delivery of nutrients to the affected area. The area receiving little or no blood flow is known as the ischemic core and is comprised of cells that die rapidly. Surrounding this ischemic core is an area of reduced blood flow called the ischemic penumbra. The fate of the ischemic penumbra is dependent upon key events that determine life or death for cells in this region, since cells in this area are in danger but not yet irreversibly damaged. Within the penumbra, the reduction in energy substrates leads to energy depletion and creates an ionic imbalance that causes cell membrane depolarization. Excitatory amino acids, including glutamate, are released in pathological quantities. Energy-dependent reuptake of glutamate is diminished, allowing for greater accumulation of the amino acid in the extracellular space. Continual activation of glutamate NMDA and AMPA receptors contributes to intracellular calcium overload. Within the cell, the increased calcium activates a series of enzymatic reactions, including the activation of phospholipases, which degrade phospholipids, causing the release of arachidonic acid, the production of nitric oxide by the calcium-dependent enzyme neuronal nitric oxide synthase, the creation of xanthine due to the degradation of adenosine, a building block of ATP, and the disruption of mitochondrial function. Each of these reactions caused by the increased levels of intracellular calcium leads to the creation of reactive oxygen species, such as hydroxyl, superoxide, and peroxynitrate radicals. These free radicals ultimately cause cell damage through a variety of irreversible chemical reactions that lead to membrane leakage, DNA mutations, and dysregulation of cellular processes. As the chemical reactions continue, the damaged cell exhibits aberrations in ion homeostasis, cellular signaling, and gene expression, all of which can result in ischemic cell death. In the mitochondria, free radicals result in the formation of mitochondrial transition pores, which play a role in apoptosis. Opening of these pores leads to disruption of the mitochondrial transmembrane potential and the release of molecules, including cytochrome C, which ultimately may result in cell death. The open pores may also continue to enhance the release of calcium into the cytoplasm, furthering the production of mitochondrial-mediated free radicals, leading to necrosis. Consequently, ischemic cell death may be due to both apoptosis and necrosis. Reperfusion of an area affected by ischemia may occur spontaneously or through the use of clot-dissolving medications. Although reperfusion is desirable, the renewed oxygenation of tissues following reperfusion may lead to a burst of free radical production that may overcome cellular regulating mechanisms, resulting in additional damage of lipids, proteins, carbohydrates, and nucleic acids. Reperfused ischemic regions also show an increase in leukocyte activity. Endothelial adhesion molecules bind to glycoprotein receptors located on inflammatory cells, such as neutrophils, which then adhere to the endothelium. The transendothelial migration of inflammatory cells into the brain parenchyma results in additional free radical production and an increase in damaging cytokines, such as tumor necrosis factor and interleukin-1. These products can further the destruction of cells within the penumbra. Pharmacological interventions continue to be investigated with the hope of minimizing the damage within the ischemic penumbra. Neuroprotective agents may be directed at several different targets within the ischemic cascade that lead to cell death. In the near future, the advancing knowledge of the ischemic process may be translated into new, effective treatments for stroke. Until then, the incredible voyage of discovery will continue as we work together in this challenging therapeutic area.